Great. I'm gonna go get some coffee before the next session. Very nice to meet you. Yeah, I'll say hi to you. I'll see you.
And do you want us all up here at the front at the same time?
Like
all sitting here,
think,
yeah?
I think
From the
that would
beginning?
probably be the easiest, don't you? Because then you have
Sure.
to get up here
Yeah, how do
with
they...
the curtain and everything. There's nothing here.
Hello, Abby.
Good morning. I'm sorry. Okay, very good. Nice to meet you. So, you be here for this session?
Yes.
Oh, very good. Thank you. Great.
Maybe in the future, we may ask for your opinion.
Oh, sure.
On the topic.
That would be great. We'd be happy to do that. Happy to interact with you. Sure. That's interesting. So how does it work?
Who's the moderator?
I am.
Yeah, absolutely. Yeah, no, today. But yesterday.
In order,
Ah, you're here. How are you? Good to see you. Okay. Good. You're all right.
So basically, like, should I reach out now while I'm in staff?
recorded.
Okay.
Please try and stick to 12 minutes. There's a timer right here. At 2 minutes, right at 2 minutes before the time is up, it'll start flashing yellow.
So we've got a little bit of a different timing thing going on.
You have 20 minutes?
Yeah, so the first one is, one is longer.
So it's like yesterday.
Yeah.
The first sessions were 12 minutes.
Yeah.
These are 20 minutes. Okay.
Yeah.
So I'll still do the 15 minutes and then 5 minutes sum up. So start wrapping up at 5 minutes at questions and answer. But please have them try and stick to the schedule.
Okay.
They're really kind of.
Yeah, I noticed this last one really hung on forever. Okay, so now what do I do to switch the?
So nothing to switch it. I'll take care of the
Okay.
switching.
Okay.
The only thing that they'll need to do is forward, backwards, and laser.
Okay, cool.
Mm-hmm.
All right. Thank you.
Yeah. Other than that,
Okay.
have a good show.
Okay, thanks. I'm counting on you to help.
Absolutely, I got you. They'll be up there. As soon as we meet them, they'll be up there.
Okay, good. Okay, guys.
And the problem is, I was having real problems with it yesterday, and it was, even when you put it on the fry, it sort of leaves a little bit.
Hi. We figured it out. We figured out what the German was. So there's one like this downstairs, and they work with treatments. They were on the same thing.
I don't even check my sheet. It was very interesting.
Is it gone? No, they would never mind.
Hey! Hi. Good morning. Good morning. What am
I will be staying there. I will be staying there too, but it's still right here. And wait for some 20 minutes altogether.
Yeah, so you guys, you and Mom do whatever you figure out. And then, and then you can, just say, okay, we're ready to open it up. I'm going to send you my whole discussion. And then after you and Mama do finish what you want to say, you can say, we're ready for our discussion now, and ask people to please come in. Yeah, I don't know. I think she was working really fast to see if we could put it up at the end, but yeah, I don't know. Okay. All right, shall we get started? Thanks everybody for coming to session 72, I think it is, which is on the pathway to deployment of gene-drive mosquitoes as a potential biocontrol tool for elimination of malaria. What we're going to do here today is review a recently published study that considered whether mosquitoes modified with low-threshold gene drive could be developed appropriately for use against malaria, and if so, what resources and activities would be needed to ensure their safe, ethical, and efficient field testing and implementation. This took as a case study, wrong way. As a case study, gene-drive strategies for control of malaria transmission in Anopheles gambiae mosquitoes, which, as everyone knows here, is a prominent malaria vector found in Africa. The study looked only at self-propagating forms of gene drive, and in particular, what's been called low-threshold gene drive, which is intended to establish and spread from low initial release frequencies or even rare introductions. The study took into account strategies that are aimed at population or replacement or population modification approaches, where they're trying to limit mosquitoes' ability to transmit the malaria parasite, and also population suppression or population reduction approaches, which are aimed at reducing overall numbers of the targeted mosquito species. And as probably everyone in the room knows, proof of principle has been achieved for both of these strategies in the laboratory. Now, why did we focus on specifically on low threshold drive? Well, I think this graphic, which our colleagues from the New Partnership for Africa's Development like to use a lot, goes a long way to answering that question. It just shows very remarkably what the actual range is of malaria transmission on the continent, and it goes a long way to explaining why delivery of conventional control tools has been so challenging. So theoretically, low threshold gene drive could provide a tool that would only need to be delivered simply and infrequently, and it could provide cost-effective and durable protection with no need for human behavior change. And in fact, modeling has suggested that situations that are challenging for traditional vector control tools actually can make it easier for gene drive to succeed. So in order to address this very important and comprehensive question, we assembled a multidisciplinary group of international experts who worked with us for over a year looking at this issue. And we brought in additional experts to help address specific topics that arose as we thought about this entire pathway. And then when the draft recommendations were completed, we had them reviewed by a group of external reviewers that had no association with the project. So all in all, more than 60 people contributed to the development of these recommendations. The recommendations were built on relevant prior guidance. Darn, keep doing that. Can we go back there? I'm having trouble getting back. Oh, well, anyway. They were built on relevant prior guidance, including the WHO guidance framework for testing genetically modified mosquitoes, which was published in 2014 and described a phased pathway for testing of GM mosquitoes with systematic assessment of safety and efficacy at each step. That's this one. And then also the Gene Drives on the Horizon report that was issued by the National Academies of Science, Engineering, and Medicine in 2016, which provided general principles to guide responsible practices in gene drive research across many applications. So the working group took into consideration what we felt was a very important need to move beyond the discussion of risks and benefits in a very general way and talk about the specific risks and benefits of a particular application of gene drive technology. So their charge was to envision the range of requirements for testing and implementing gene drive modified Anopheles gambiae, specifically in Africa, and that included consideration of containment requirements, efficacy and safety testing, monitoring, ethics, stakeholder engagement, policy, and regulatory issues. They also needed to identify capacity and infrastructure needs that would have to be addressed to prepare for this pathway. And it was important to understand gene drive modified mosquitoes as a tool within an overall strategy for malaria elimination, which went a long way to thinking about some of the problems that people have raised in the past with this technology. And finally, the intention of the recommendations was to provide guidance to support planning by everybody, by developers, funders, regulators, and policymakers. So today's speakers are going to be, oh great, having a bad time here. Steve Lindsay, who was a member of the core working group, and he is going to give an overview of the recommendations. And then Willie Tanui, who is the former head of the Kenyan National Biosafety Authority, will talk about the regulatory and risk assessment challenges. And Claudia Emerson from McMaster will be talking about community and stakeholder engagement issues. And then my co-chair, Fred Rosakumu, and Mamadou Koulibaly on the end there will be sharing their thoughts about the development of this technology for Africa. And we're going to hold the questions until after all of these talks have been finished, and then we'll have an audience discussion session, which Fred Rose will moderate. And with that, I'll ask Steve to come up for the first talk.
Whilst I'm waiting for my presentation to come up, good morning, ladies and gentlemen. It's a great privilege to be able to present this for a large number of people that are involved with this exercise over the last couple of years. I just want to make sure that you folk at the end can hear me okay. Is that coming through now loud and clear? Excellent. So, okay. I always get very relieved to see my slides come up rather than just standing here wondering what I'm going to say. Pardon?
Is this your presentation?
It is. Looks like it. Thank you. There's usually a picture of my mother on the second slide, so I can see that. So, if you can give that full screen, we can go. It's okay. So I'm all right. So, right, good. So my job here this morning is to overview what was decided in these guidelines for the release of GM mosquitoes. So the exercise here is to go, what is the pathway of progress from the lab to actual final deployment in the field. Okay, so the background. So the exercise really was thinking about malaria control in Africa, and specifically the control of Anopheles gambiae. And really, the whole exercise was gauged around the idea of making sure that this technology would be safe and efficient before final field testing and deployment. So I'm pressing and it's not moving forward.
Let me try and close it and reopen it.
Okay. That's it. Beautiful now. Okay, great. Okay, so we considered two genetic methods of control. One is suppression, and the other one is population replacement. And I'll explain what that is in more detail in a minute. But first of all, this pathway of development didn't come from nowhere. It was based on WHO's work from the 2014 publication, which I can show here. This is a 4-phase procedure, and it starts with the lab studies. First of all, it then moves to a second phase of field testing in natural settings under strictly confined conditions, before moving up to a third phase, which is about entomological and epidemiological outcomes, and then finally deployment of with continued surveillance. But the world has moved on very rapidly since 2014. There have been some major inventions, if you like, particularly with CRISPR-Cas9, which has transformed the scene. So this is one of the reasons to take a closer look. So the two basic mechanisms for the release of GM mosquitoes is either replacement or suppression. Now, if you look here at the top slide here, the black mosquitoes represent the wild population, the grey mosquitoes, the GM mosquitoes. And what you can see here is that progressively, with the drive mechanism, that over time, the genetic construct spreads throughout the whole population. Now, the important thing here is that you then have refractory mosquitoes to malaria mosquitoes, to malaria, hopefully, in the end. So you still have mosquitoes in the population. This is very different from population suppression, when the gene drive mechanism suppresses reproduction and causes a collapse in the mosquito numbers. So in essence, you may actually result in elimination of the mosquito on a very wide scale. So the spread of this gene drive requires consideration of a number of aspects. First of all, if you have escapees in the field, they may establish themselves in surrounding environments. And through sexual reproduction, that construct can spread. Importantly, it may spread so rapidly it can go across borders. So you start in country A and it has potential for moving across to country B. So that is very different from a number of other vector control interventions. Finally, you may have the position that the modification persists in the wild population. So what you're doing is very different from previous experiments. So this was, again, a reason that we need to revisit the 2014 recommendations by WHO. And I would say that in our discussions, we did work very closely with WHO, and we had members of WHO past and present. So here's my own personal piece, because I think a lot of people are concerned about, well, if we go for the population suppression and we eliminate Anopheles gambiae, does it matter? And here's my personal view, not the view of the team. I want you to remember that Anopheles gambiae is humankind's greatest killer. When I started working in malaria 30 years ago, there were 2 million children that were dying each year from malaria. Today there's 400,000. Most of those are in Africa, and that's 400,000 too many. We have eliminated mosquitoes from Brazil and Egypt, Anopheles arabiensis. And don't forget that insecticides have been deployed on a massive scale, and we've killed trillions of Anopheles gambiae, and nobody has, as far as I'm concerned, has worried too much about that. Ecologically, it is rare in nature. You actually have to go and find and search for it in aquatic habitats, and you have to be a bit of a hunter. But it is beautifully attuned to finding us, so it occurs in large numbers inside the house. Now, I can only think of one example, there may well be others, of an animal which solely relies on Anopheles gambiae, and that's a small indoor spider in East Africa. I think that spider may go as development improves and the number of houses and house improvements, that spider will find it less favourable to exist. So overall, I think if you actually remove this mosquito, I think it would have little impact on the local ecosystems, but only for this mosquito species, because it is so beautifully adapted for people. So now here's the pathway that was developed. We start, of course, in the lab, and then we move down this pipeline from small-scale isolated releases to larger-scale, small-scale releases. Then finally, when we have confidence in what we're doing, in scaling up, we do large-scale open releases with epi outcomes. And then finally, massive deployment in an area for control. Now, part of the thinking of the group was enforced by people which had a wide range of experiences. And so these were not just molecular biologists and entomologists. They were people from the biocontrol environment, where you're releasing new animals into new environments. And so we're learning from the methods and mechanisms that were established for doing that. And secondly, the release of genetically modified plants and the sort of obstacles and challenges that presented, as well as expertise from people that understood and designed clinical trials for vector control tools. So these are the highlights then. And so we're looking at the safety, efficacy, containment, monitoring, engagement, and regulatory aspects of this. So it's not entirely about thinking about the effect of the mosquito on malaria transmission. So this is providing evidence for a whole bunch of people, from researchers to regulators and funders as well, because this is a large-scale community that we need to think about. So in the laboratory studies, the recommendations are we should avoid premature release into the environment, and the sort of containment facilities that are required at arthropod containment level 2, which requires stringent physical control. And the candidates for field testing must show efficacy and fitness with the product claim. And in these early stages, it's really helpful to have modeling to support the outcomes because they aid with strategic thinking. But of course, all models are wrong, but some are useful. So they're not the real life situation. So partnerships and engagement, I think, are really critical from an early stage, which is why I think this publication is so important. If gene-derived products are developed from outside Africa, there must be strong partnership with African scientists and institutions from the beginning, in the early phases of development. And the partnership really must be in a spirit of co-ownership and co-development. This is a teamwork. And in-country institution must take a lead on regulatory and stakeholder engagement interactions. Now, this graphic shows you that just to sort of gives you a taste of the sort of involvement here. The research team is at the center of these bubbles, and this is both in-country and external research. But there are a lot of players involved in this, and probably many more than are shown here. So including the regulators, the ethics, NEPAD, which is the New African Partnership for African Development, ministries of health, NBA, that's not the basketball, that's the National Biodiversity Association, the media, and most importantly, the communities. So there needs to be an openness here. There needs to be an openness amongst the researchers themselves, and to avoid any rush to be the first, there needs to be sharing of where we are and with data sharing as well. This transparency is utterly important to earn the confidence of the public and reduce anxiety. And I actually think that if you want a better guiding light for this, the folk that have been doing the work on Wolbachia in infected mosquitoes and the releases have been absolutely exemplary. They spend a considerable amount of their time in working with communities and a huge amount of their budget. And I think that really is important, absolutely vital. Okay, so it requires appropriate processes and platforms for broad sharing of information and make records of any releases, whether it's accidental or intended, available. Share best practices, publish baseline field data, and make public monitoring results. And this is something that we're asking the funders to think about as well. Important, those people that are going to support these developments. So where do we do it? Whereabouts in Africa? What are the things we need to think about? So first of all, we need a suitable biological environment. It must be targeting, don't make it too difficult. What's the major species there? Is it an Anopheles gambiae? Is it sensus stricto, Kaluzi, whatever? And presumably you want an area where malaria is not too low and not supersaturated, so somewhere in between. There must be no obvious social or political barriers, no widespread public or institutional opposition to similar technology. There must be support from the government. And there must be also the legal and regulatory framework in order to help progress these interventions. So it's important, it's not just doing the experiments, it's far bigger than that. And one needs to move forward progressively, carefully, and slowly. So the first situation is when you step out of the lab to go in to do small-scale releases. So it must be absolutely, well, as absolute as you can get, confident that your product is not going to do more harm to human health and the environment than wild-type mosquitoes of the same genetic background. Also, that resistance won't arise too rapidly to allow for positive health impact. And releases should not begin until funding is secured really down the pipeline, because once you start, then there is I think an ethical reason for carrying on, and that must be a long-term commitment. Particularly, and the major consideration, of course, is the safety of the participants. Risk assessments are going to form an important part of the process, and these must be independent, particularly for that first step, moving from the lab to the field, and looking at both social and technical impacts. There's also a role here for self-limiting constructs. So what we mean here is when you remove the drive mechanism, so the mosquito may only survive for a few generations or single generations. So this is not the final product, but it's a sort of halfway house to provide people with the confidence that this is safe. And it allows you to do all sorts of things to see how the mosquitoes survive in the field, but also it's an important training exercise. This is a massive learning process. Monitoring evaluation is key, and this needs to be thought through well in advance. And we need to develop new procedures for picking up escapees, for being sensitive to areas in which we're doing the releases. This must be agreed before the trial, and ideally it should be, again, done independently from those people that are doing the releases. And plans must be in place for remediation if there is pullback. So it's not just release and then close your eyes, it's following this through. So ecological surveillance is also required, and this must be done well in advance. So when one comes to do those first releases, what does it look like? Well, before you do releases, you've got to know really what's going on in the environment. So at least 2 years baseline entomological data and ecological data for at least 2 years is the recommendation. And whilst we're not being prescriptive, for some people are happy to use semi-field releases. So this is a semi-field site, and what is effectively it is a large greenhouse, where you can do releases under controlled conditions. But even then you need to be concerned for escapees or whether accidental or otherwise. But again, this could be useful for boosting support for the regulators and ethics committee that what you're doing is safe. So if you're going to do this sort of release, these sort of experiments must be done near sites where you're expecting to do the first field releases anyhow. But it also requires monitoring for escapees around the outside, just in case there are releases. So then we move on to the small-scale releases, looking at entomological outcomes primarily. So these experiments are done really to better understand the mosquitoes themselves and the drive mechanisms. So it's related to the rate of spread of the transgenic construct and the ability to suppress or replace vector populations. And it's important as well to monitor the construct stability to see whether it changes over time. So there's technologies that have got to be developed and scaled up in order to do that. It's important also to restrict environmental exposure. So these first releases need to be done either in island situations or in situations where they may be remote from, maybe surrounded by dry land and desert, but relatively isolated populations. It's important, again, the safety concerns is paramount to observe what's going on, and this may be be demanded, is likely to be demanded by regulators and ethics committees, and particularly looking at the interactions of the gene constructs with other mosquito species. So there needs to be, I think, when it comes to the environmental impact, there's a lot more thinking that needs to be done to try and to decide what are the key measurements that one needs to make, what are the organisms we're most concerned about, and to develop technologies for picking up those adverse interactions should they occur. We're not expecting them to occur. We start with the principle that this is safe before we do the releases, but we need to expect the unexpected. Any evidence for adverse impact would be a no-go stop, so for further testing. It may be important, I think it will be important, to do multiple releases in different sort of environments, because it may work well in one area and not the other for various ecological reasons. It may be too hot or whatever. Large-scale releases, then here we're looking at the impact on disease transmission and spatial dispersal, and particularly what we're interested here is not the effect on the mosquito, but is it reducing malaria in the local population, infection and incidence. And yeah, need to consider other malaria vectors. We need to monitor the effect on population structure and on non-target organisms. So that's fine. And then we go to implementation, and this requires decision-making to go from regional deployment to regional deployment. So a very important aspect of these releases is regional conversations, avoiding surprises, and because it's a wider issue here. If one goes to scale up, that decision is based on the efficacy of the construct, the safety, the perceived benefit, acceptability, and the cost. This should be not considered as a standalone intervention. This is integrated vector management. It's additional to the other vector control tools that we're deploying across sub-Saharan Africa. It's not a cheap fix. It's an additional tool. It's very important to WHO to play a leading role in this, and I'm sure that they will do, because it requires conversations across countries, across regions. It requires long-term planning for the future. Business models need to be developed, engagement with governments, quality control and standards need to be thought through. And after implementation, surveillance doesn't stop, it carries on. And again, WHO here has a key role because of the scale of these operations. And it's going to require considerable amount of injection of capital to continue to do the surveillance to make sure that it's working. And that includes not just watching what's going on with the construct and seeing whether there's resistance developing, but potential adverse effects on the ecosystem. And as with all interventions, resistance may well occur with all interventions. So what are the resources? So what do we need to make this work? More sensitive monitoring tools for escapees, standards and best practices, information and data sharing, support for risk assessment, and really thinking through what is required. Support for effective stakeholder and community engagement, opportunities to build capacity in regulation of modified insects, regional harmonization, quality standards for manufacturing of these living products, and mechanisms for oversight and management post-implementation, and the development of assays for product performance failure and systems for reporting performance failure. Finally, the coordination here is key. From the perspective of product development, inappropriately conducted field trials have the potential to negatively impact the future success of other gene-derived products, which will undermine the community, stakeholder and public confidence, and to contaminate the regulatory and funding environment. Here is a plea for people to go slowly, carefully, because what you do will affect this the series of interventions, which has potential to make a great difference to malaria across sub-Saharan Africa. We're all in this together. We don't need the woodpecker picking holes in the boat to make it sink prematurely. Thank you for listening.
on to Willie Tanui to talk about regulatory and risk assessment issues.
Yeah, good morning, everyone. It's an honor to be here this morning, and as my talk is being prepared, I wish to state here that, yes, I'm going to give you a short overview on regulatory considerations for gene drive. And most countries do develop their laws and regulations based on international agreements. And two important agreements here are important for gene drive. We have the Katakana Protocol, which was a product of the CBD, which was adopted in the year 2000 but came into force in the year 2003. And that protocol has been used widely by various countries to develop their own biosafety laws. And it was recognized that the Cartagena Protocol did not include aspects of liability and redress, and therefore a supplementary protocol was developed, and it came into force this year in March, actually March 5th of 2018, and it's known as the Cartagena, Nagoya Kuala Lumpur supplementary protocol. And the Cartagena Protocol is a governing tool for international based. It's supposed to enforce governing of transborder movements of GMOs and concerned mainly with the potential adverse effects on biodiversity or human health. And 171 countries so far have agreed to this instrument, including many African countries. And the Cartagena Protocol provides for designation of a focal point, and also the establishment of national biosafety agencies or authorities in this case to work on matters of GMOs and including gene drives. And the Article 17 of this document has provided for establishment of biosafety clearinghouse. This is an internet portal whereby countries do provide information about their approval processes and what they have approved in different countries. And the instrument also provides for transboundary movements and use of border measures, to prevent adverse effects from moving to different countries. And as you are aware, gene drive in this case are likely to be uncontrolled, and therefore matters of international considerations are also be put in place. And the document provides a recommendation that there should be mechanism for regional harmonization, and authorities also be consulted before testing. And in the African continent, we have the NEPAD, the African Union, that is responsible for oversight in different countries in Africa, and therefore they have to be consulted and work with the team to support the regulatory development in the region. The role of WHO also has been discussed by my colleague. And in most countries, many countries now have developed their laws and they have experiences on GM plants. And so for gene drive, these are the same authorities that are going to play a role. And the regulation in this case will involve both biosafety and health regulators. The biosafety agencies will be expected to be the first point of regulatory contact, and therefore should be approached so that they can become familiarized with the technology and also to support the process. Health regulators also may come into consultation in later stages, and also the environmental regulators will also be important at later stages. So this is a first approach that you've been familiarized with, and safety is a very important consideration during every first process. And For laboratory studies, for first movement from laboratory to the field, the criteria will be recommended. Recommended will be that the gene drive will have no harm to human health than the wild mosquitoes, and also providing no harm to the ecosystem than the conventional vector control interventions. In terms of biosafety, the instrument basically strives to provide for maximum safety and approval, at all stages, and also providing for opportunities for reduction of adverse effects to humans, animal health, biodiversity, and social and socioeconomic considerations. You realize here that risk assessment is very important before any decision-making process is made. It is important to ask yourself, why are we doing this risk assessment? Who will be doing it? And risk assessment will also differ if you are dealing with a construct that is suppressing or causing replacement. For consideration for field testing, it's important to think in terms of monitoring for adverse effects, and also to determine what happens, after the trials ends. During implementation stages, the WHO pre-qualification process will be very important here. It's also important for the developer to do a cost-benefit analysis, and ensure good manufacturing and quality control procedures. Registration also will be very important from the regulatory standpoint. How is it done in your different countries? How do they do post-market surveillance? Issues of liability and redress, should be very important. Should the product require insurance? And so this may involve different actors as you move down the pathway. And I'll speak briefly about the regulatory gaps and challenges in Africa. I've mentioned here that most countries have developed their biosafety laws based on the Cartagena Protocol. And so far, as you can see several countries are yet to develop their own biosafety laws. Most of the region have yet to develop laws. A few countries have moved on to commercialization. That is Burkina Faso, Nigeria is also registering, Sudan, and Ethiopia. Other countries are also in the pipeline. But basically, most countries have experience of GM crops, and therefore, issues of gene drive will present new regulatory challenges to most of these organizations. And the good part is that many countries are now developing their guidelines and addressing are addressing their laws so that they can also provide regulation to gene drive. And therefore, there will be need to consider building frameworks and relationships in different countries to ensure transboundary movement. And NEPAD is doing a great job in this, in mobilizing people to move forward with the technology. So far, the NEPAD, as you have heard, is the regulatory arm of the African Union. It has been conducting several regional consultations. They have also established a high-level panel on gene drive, and recently the team developed a document that makes case for gene drives to be considered additional intervention to achieve African Union target by 2030. And lastly, efforts are currently being done in different countries to develop laws, guidelines, based on their limited experience on GM crops. And there's an African saying here that these are small efforts that are going on through the pathway document and other efforts that are being done. And if you think actually you are too small to make a difference, you haven't spent a night with a mosquito. I believe most of us have spent a night with mosquitoes. And it's possible. And as our departed leader mentioned before, we seek our inspirations from him. It's always possible when it seems impossible until it is done. Thank you very much for your attention.
There she comes. Now we'll hear from Claudia Emerson on community and stakeholder engagement issues.
Great. Thank you, Stephanie. Good morning, everyone. I just wanted to point out, I've noticed a few people standing at the back for a long time, just to let you know that there's empty seats at the front in case you're getting tired. Okay. So I'm tasked this morning with presenting on the working group's recommendations for community and stakeholder engagement. And my sense is that if there's ever an audience of people who appreciate the value and importance of community stakeholder engagement, it's probably the folks in this room and indeed at this entire meeting, because I think we all know how valuable it is for global health research, for any research, in fact, that involves human subjects. And in large measure is because we are dependent on communities when we go in and the work that we do, that there's an important relationship that's built, a trust relationship that's built in working together with communities. And a lot has been said and written about community engagement in different contexts, in social science, in medicine, in health, and a lot of good work around what works, what doesn't work, lessons learned, best practices. And I think we can say at least two things about community engagement and stakeholder engagement, that it has both an intrinsic value and it has also a practical and instrumental value. So from the very practical side, community engagement is essential for working together with the communities and understanding what their values and concerns are, and ultimately to reduce the risk that a particular technology or intervention is going to be disruptive, or that the research is not going to have any value to them. And of course, there's always reputational risk for the researchers, for the sponsors, for those who are involved, all of the actors who are involved in any kind of research enterprise. It's important to understand what that is. And so community engagement is at least one way to understand what those risks are and how we can work together to overcome them. But more importantly, I think community engagement has this really intrinsic value. So this fundamentally about relationships with people working together to solve a complex problem, and trust is critical. So community engagement is essential for building trust. And what's going to be important, obviously, is going to be culturally appropriate engagement, which is reciprocal. So this is just not about going into a place and understanding needs, but it's a learning and sharing in good community engagement endeavors. The community is involved in shaping the research, the questions, the outcomes, and this is critically important for a good engagement process. So in terms of the recommendations that came out of this report, there are two things I think that are important in terms of the language and the terminology and how we think about community engagement. So one of the first questions is going to be who engages with whom. And an interesting shift happened from the WHO guidance framework of 2014 on testing of genetically modified mosquitoes, where the language of community engagement was related to the project team, the host community, and third parties. And then you fast forward till 2016 and the National Academy's report on gene drives, and the notion of community engagement is talked about in terms of communities, stakeholders, and public. So there's a little bit of a shift in terminology, and this is actually important because it says something about how narrow or how broad we're going to define a community, and also the implications for understanding interests and which interests ought to be prioritized in any kind of project. And so the community is defined in the National Academy's report as a group of people who live near or potential field trial or release site and that have immediate interests in the project itself. And this is a little bit different from how people understand communities in other sort of guidance documents. So for example, the Council, the International Council of Medicine, SIOMS, describes community as not just being those who are immediately impacted by research, but could be much broader. So it could be, for example, all of us sitting here, the global community. So I think the important message here is that people have very different understandings and concepts of what a community is. But for the context of a gene drive project where there's going to be a broad, so this is going to be a global, stakeholders are going to have, global stakeholders are going to have an interest in this. One of the key things to remember is that the community, the one that's going to be involved in, say, those initial first releases and tests actually have very critical interests. And so there was a desire to kind of narrow down that definition. So then a stakeholder is going to be a person with a professional or personal interest sufficient to justify engagement and may not have a geographic proximity to the release site. And so one thing we can say is that stakeholders are obviously going to include communities, and it's going to include those much broadly. So basically anyone with a stake or an interest in this kind of project, and that includes all of us here, is going to be a stakeholder. And then of course publics, this was important, to designate publics, a shift from third parties to sort of different publics, because you're going to be, according to the National Academy reports, different publics, and those are those who have lack an immediate connection to the project, but nevertheless have concerns, hopes, fears, and values that contribute to decision making. And this is both folks that support projects and those who are have concerns and are opposed to them. And so a key thing to know is that the composition of these groups is going to change as the project evolves. There is going to be different obligations to each of these groups and in terms of what that engagement is going to be like. So the depth and breadth of engagement is going to change depending on who you are engaging with and what the goals are. But a key thing that's going to be common for all of them is that the interaction is going to be ongoing and iterative. So it's going to start at the beginning of the project and it's going to go all the way through. So I mentioned at the start that engagement has both intrinsic and sort of an extrinsic instrumental value and also an intrinsic value, but two other things that we can say about community engagement. One, that it's a science, and so it's a science in and of itself like any other science. And it's going to require a lot of sort of focused research and expertise in order to do this right. And so one of the things that the group was very focused about stating in the recommendations was that engagement should be context specific, culturally sensitive, and from what we've learned about best practices and research in the field is that it's best undertaken by locally known and respected persons. We also know that it's going to be an ongoing process, and one of the key things, and you all saw sort of the pathway that Stephanie laid out at the beginning, the engagement is going to start at the beginning and follow all the way through, including to post-surveillance activities. It should involve experts who are familiar in both the methodology and the practice, such as social scientists, ethicists, and others who can be involved in the development of a plan at the start and in its implementation. Another key piece, so because it's a science, we ought to monitor and evaluate, and one of the recommendations the group made is that be able to capture sort of those lessons, what works, what doesn't work. Is there anything unique about engaging in this area when you're releasing a sort of first-in-class novel technology where the potential risks and benefits need to be ascertained? And sort of an important message, and I think colleagues who work in this space are particularly, love to emphasize the idea that community engagement should not be confused with public relations, communications, or advocacy, though those things can be tools to engage. So the other point is that community engagement is an ethical obligation. So it is both a science and an ethical obligation. And this is pretty clear, and everyone here works in the field of public health or global health, and we know that it's important to engage with those who are going to be impacted and affected by the research that we undertake. And indeed, it's one of the requirements also in a lot of international guidance for human subjects. And so The ethical obligation here is about fundamentally about respecting persons and communities in their way of life, and that the research does not disrupt it in a way that leaves them worse off, right? So it's important to understand the needs and values and concerns, including for trial and post-trial activities. And so the post-trial activity actually is quite important because you don't want to go in and exit. You leave a footprint, and so it's important to know what those expectations are once you exit a community. Demonstration of respect is going to be dependent on the breadth and depth of the engagement. As I mentioned, it's an iterative process. So depending on which of those groups you're engaging with, the community is obviously going to involve a deeper level of engagement than, say, with the public, where you may just be sharing knowledge or information about the kind of research that's going on. You want to ensure that the value, that there is both ethical and social value, and these are important ethical requirements in all research involving human subjects. And I believe Steve talked about transparency throughout the process. And so this is important to think about. So with other researchers, with regulators, this is probably a mechanism that's going to happen through data sharing. But trying to be transparent with, say, publics, it's important to think about what kind of mechanisms you're going to use in order to be transparent with those who have interests and want to know about the research. We know also that as an ethical obligation that once fulfilled, community engagement actually improves the quality of research. So it improves the experience. A lot of research studies have shown that it improves the experience of research participants and of researchers and those that are directly involved with it, with the research itself. But it's also helpful in coordinating other studies and things that are going on. So this is a space where maybe there's a handful of people working on this type of work. You want to be able to share lessons learned. Maybe a particular community and adjoining communities will be jointly impacted. So this is one way through engagement that you can coordinate what's going on. And then, of course, both Steve and Willie talked about the importance of engaging with relevant oversight bodies, and that level of engagement is going to be different, but should also start at the beginning. So with your local, national, institutional, regional bodies, so biosafety committees, ethics committees, with ethics committees particularly important, since this is going to be the first time that we try anything like this. It's going to be the first time that they see a research project like this. So it's important to be engaging with them early and in leading up to this, to be building capacity about what sorts of things are we looking for when we're reviewing these types of research protocols. And so in thinking about how we engage with these different groups, there's an overlap really in terms of the development pathway, so from beginning to the end. There's engagement at the level of exchange of knowledge and data sharing, and also with communication and outreach activities. And so in supporting the development pathway, one of the first recommendations that the group made was around engaging an independent ethics advisory committee of external experts to support the project right from the beginning. So this is getting folks who are separate and apart outside of the research group, independent experts who can guide, sort of raise and understand what might be some of the ethical issues, both enablers and barriers to the project, and think about how these start right from the beginning, from the time you have the idea all the way through to your post-implementation surveillance. And actually, some of the groups that are involved in this research do have these kinds of independent ethics advisory committees supporting their work. The other recommendation is that the engagement activities need to be tailored to each phase of the product development pathway. And so we've already heard that engagement should be early, it should be sustained, but part of that tailoring is understanding what's needed at the beginning. And so in our group, there were discussions around whether, for example, at the level of the lab, or even in the sort of the cage studies, once you go into community, whether we could invite community members to come and tour the facilities and see what's going on, how the mosquitoes are being reared in the insectary, what kinds of things are going on, give them an opportunity to sort of view what's happening, understand questions. And so that would be sort of a very tailored and specific activity that we would do right at the beginning. And you could imagine all sorts of things that you might do all along the pathway that are specific to this to address any kinds of concerns that people are expressing, questions that they want answered. And so this kind of tailoring is going to be critical, particularly to this. And it's something that we don't often do in sort of your standard sort of vaccine or drug kind of testing where you have engagement processes, but we don't necessarily invite people in and say, do you want to tour my vaccine lab, right, and see what's going on. the other important part of engagement, and this will be at all levels, is for sharing knowledge and data that comes out of the project. And so one of the key recommendations was to keep open and accessible records of any accidental or intended releases, make all risk assessments publicly available so people know what's going on, share field data with malaria research and control communities. So this is at the level of the stakeholders that are in the researcher groups, consortia, and then make baseline data and modeling results available. Obviously, this is a first in class. It's the first time we're doing this. We want to make sure that everybody's learning from each other. But also part of sort of keeping those risk assessments public and the kinds of informations that we share, determining what kind of information, maybe broader publics. So just folks who read the newspaper. I mean, we've all seen some of the sort of the media published around gene drive. Some of it can be quite positive and probably too far off around, this is the panacea. This is what's going to solve, vector control or vector disease issues, and then we see the sort of the other side of it and concerns around Franken-Mozzies and other kinds of sort of very hyped up negative media around gene drive mosquitoes. So I think it's important to have sort of public facing information shared for the consumption in that group. And that's the part around communication and outreach. And so part of that is to consult also, so engagement with local authorities on the development of plans for that broader public, create materials that are appropriate for engagement, that explain clear language, what the audience needs to know about this type of science, including for those who disagree. So folks who are very opposed to GM obviously are going to express very strong views. It's also important to engage with them. We recommended consulting with sociologists and linguists to develop the necessary vocabulary. So some of this terminology is not exactly translatable in all languages, and it could be very difficult to explain to people what a gene drive is. And translating, obviously, materials into local language and idioms will be critical. So we want to ensure consistency in communication and reaching out to the different stakeholder groups. And the group discussed the importance of having somebody who has experience in science communication, or somebody who can undertake this training and be able to sort of deal with some of the questions that arise. The question of whether obviously a scientist, and I saw it within the program, there's been a lot of great sort of side meetings here around communicating with the media and how do you communicate the science. Scientists do have a responsibility to communicate their work with the broader public, and so a key piece of this is engaging with the media, who, as I already mentioned, can sometimes present very skewed perspectives on something like a novel science like gene drive. And so it's important to have these plans that can engage with them in a very clear manner. And lastly, be ready with FAQs, anticipate confusing and controversial messages. Be ready that there's going to be folks who are going to be strongly opposed to this and will be very vocal about it, and we just want to be ready for that. So thank you very much for that. Thank you. Stop there.
Thank you very much, all my colleagues. I hope that the audience, our friends in the room, can stay a little longer for the forthcoming discussion session. We will now have a brief session to take country perspectives. Very, very unfortunately, our friend Dr. Abdoulaye Diabaté has not been able to join us. But we have with us Mamadou Coulibaly. He's from Mali. And in Mali, it's one of the three African countries at the moment where our partners Target Malaria are already running a program that will eventually end in the release of gene drive mosquitoes for malaria control. My name is Fredros. I work for Ifakara Health Institute in Tanzania. And I will also provide 2 minutes of what we could call country perspective. But I really just think something that we could say was the views from the continent. So, and again, Steve did clarify that they were his personal views. These two are my personal views. First, congratulations and thanks to Stephanie and the team from FNIH who helped us to organize this entire group and really make sure that we could bring together the different experts and look at this important subject in detail. The work that we do at Ifakara at the moment is not in any way related to the molecular aspects of gene drive, but it's really to provide support from an ecological perspective of what will happen in future. So if you think about it, in the end, assuming that the construct designs are successful and the regulatory approvals are there, we will need to make sure that the genetic constructs that we release can actually spread from one place to another. And we have to guarantee that, first of all, by really studying the gene flows in these mosquito populations, and also being able to rear these mosquitoes in a colony. Because if you cannot create a colony of these species, you will not have anything to release. And some of the, of course, there's an assumption that you can rear all of them, but not all of them are that easy to rear. So we have projects, for example, that are looking at how can you establish colonies for Anopheles funestus, for example, which is a major vector in Africa already. When we started this work, there was something very interesting that was called Phase Zero. I don't know if you guys remember this. And at that time, Phase Zero was the idea that you would have a construct built in the US or UK or somewhere, and that would be the Phase Zero of it, and then it would be sent to Coulibaly, to Mamadou, and then Phase One would begin from there. What happened very interestingly was that the group formulated this so-called co-development, which is an idea that came from target malaria, that actually recognized that you can build these gene drives from the very beginning, starting to work with the African partners at that time. So that was quite nice. Something that I would like to reiterate is the reason we are involved in this is so that we can understand it better, so we can inform our regulators better, because we also have a choice to say this technology sounds like it's so dangerous, we don't want to get involved. You know, if we do that, it means we will not have the information about how it works, what are the risks, what are the successes. So there's really an encouragement, and this has also been expressed in the Africa Union document that has been released this year, that African scientists need to really get involved in the science of gene drive. So that if we have questions, Claudia did mention we need FAQs. If we have questions, that we have people who can actually answer these questions. So these are a big reason here for our scientists to be involved and to stay involved in not just the discussion about it, but actually the science of it all. Other thing to reiterate is the multiplicity of species. I think Steve spoke about this in the beginning. There's always an assumption, and this is the most common question actually that people ask: what will happen when you eliminate mosquitoes? You know, you get asked all this all the time. As Steve was saying, we've been trying to eliminate them for ages, you know, spraying everywhere, killing billions of insects. But I just want to say that actually we have, 4,000, maybe 3,500 species of mosquitoes. 400 of them are anophelines. About 30, 40, up to 70 sometimes can transmit malaria. But in Africa, there's like 3 or 4 that are the major guys. So assuming that we have a gene drive construct, we will probably end up eliminating 1 or 2, if the elimination issue was the big deal. But if modification was the big deal, we would modify only 1 or 2. So actually, in terms of biodiversity, there's still quite a big room. So if we work closely with our environmental guys, our regulators, there's still a lot of room here to do this safely without creating a diversity problem. Lastly, it's important for us to play on the due diligence side to make sure that we are as transparent as possible. So we must not create a narrative here that gene drive is entirely good. We also must not say that it's entirely bad. So if our scientists get involved, we will be able to say these are the advantages, these are the disadvantages. As someone who comes from Africa, I am also aware that we will be the biggest, we will accrue the biggest gains. In other words, if this was actually successful, Africa Union says we want to eliminate malaria by 2030. I don't think we can do that with bed nets, with IRS, or with case management. I think those things will make a lot of gains, but there will still be quite a lot of gaps. So things such as GeneRev give us the opportunity, in case they are successful, that we can actually accelerate this process and get to that goal of actual elimination. Recognizing that there are challenges associated with this, I think there is reason to be involved. And if we discover at some point that this is a dangerous path to go, our scientists will be able to tell us. But we've got to build that capacity for those scientists to be able to say that. And this is why people like the work that people like Mamadou Coulibaly are already doing is incredibly important. And we need to support this one going forward. If we don't, we are stuck with this. So I'm going to invite my colleague Mamadou here to just give us a little bit of the description of the work they're doing in Mali, some perspectives from West Africa. And already I should say congratulations to him and his colleagues on the work they're doing. We thank you very, very much for taking the lead. We are right after you and we will support you. After that, we'll have a Q&A. Thank you very much, and let's welcome Mamadou.
Thank you, Fredros, for this brilliant introduction. My name is Mamadou Coulibaly. I am a pharmacist to begin with, but I got bitten by an insect called entomology. And since 2006, I would like to call myself a vector biologist. And I'm also the coordinator for the country for Target Malaria, the project that Fredros mentioned to you. So this is a non-for-profit research alliance that works on genetically modified mosquitoes to fight against malaria mosquitoes. So I don't have slides for you today. Maybe it's because I grew jealousy to slides. Just I want all attention for myself. Before I get to the perspectives for the country, I'd like to just do two reminders. One is about malaria burden. Steve mentioned some of it globally. So you got like more than 200 cases of malaria occurring every year with about 500,000 deaths. And the 80 to 90% of those cases and deaths occur in the sole sub-Saharan Africa. And for Mali, where I am from, there is more than 2 million cases every year, with about 2,000 deaths every year. So I don't think this is acceptable in the 21st century. And it's not because there is no tools. There are existing tools. We have bed nets, we have IRS, but I guess everybody will agree with me that those have been around for long. They are efficient, but maybe not enough to go to elimination or eradication. So definitely there is a need for additional tool for us to go to the elimination or to the eradication objectives. So for that, gene drive technology, GM mosquito technologies, I think should be considered and are being considered for additional tool to get to the objectives. The second reminder I want to bring up today is also why do we need additional tools? Sometimes we get this question, why? We have BedNet, you have IRS. Why do you need additional tools? Well, one thing is, for now, bed nets are efficient, but indoors. They are used indoors. IRS is very efficient, but again, it's indoor. And if you look at the data now, for entomological data now, you look at the biting place, it's almost split between indoors and outdoor, like almost 50%, 50%. If you collect 100 mosquitoes, indoor and outdoor, you may find like 50%, 50%. So you got the transmission also going outside. And where I'm from, Mali, people, it's a tropical area. It's very hot. So people, stay outside for a long time, up to midnight. and then you get chances to get your malaria dosage before you go under the bed. The second part is the insecticide resistance. Bed nets are using insecticides. For IRS, we are using insecticides. So there is a resistance developed in mosquitoes. So those two together, I think, trigger a thoughtful development for new tools that can be used to complement the existing tool. As Steve said, gene drive or GM are not going to be a magic bullet, but I think there could be a very good additional tool complementary to the existing tool. Now, what are the perspectives for Mali? What are we doing in Mali? In a nutshell, or just in a sentence, I would say maybe there's a need to adopt biotechnologies, including gene drive, to help save lives in Africa, not just in Mali, but in Africa, and that in a responsible way. Again, if you are to speak after many of those good speakers, you don't have much to say. They have said all. But again, I will probably come back to those ones. So what do I call a responsible adoption of those biotechnology? It's adopting gene drive or GM mosquitoes in a responsible way means to me, not saying that everything is good. The gene drive will just, on a click of the finger, make malaria go away, or it's really too bad. But that's not what I call a responsible way of adopting it. But we can accept it with all the precautions, the advantages, and acknowledging the risks, and also the benefits and the opportunities. The first thing that comes in my mind for the perspective is the regulatory part of the question. In Africa, in some of the countries like mine, we have already started building the regulatory bodies. The laws are ready, and you also have the organs to make the decisions. But everything is not perfect because there is need for training, training the regulators, to be able to make informed decisions. And so this has already started. NEPAD, I think Steve mentioned NEPAD, Danford Center. So and FNIH, I see Dave in the room. So these are bodies helping the national regulatory bodies to be effective in making informed decision. And there is another way. Mali and Burkina and Uganda, yes, we are probably starting the project, but we are not a silo. So there is also a regional regulatory text documents and policies that also are taken into account in adopting the technology. The African Union has made a statement, encouraging governments to consider the use of gene drive for malaria control, and this is a very good asset for the countries. The second pillar, which is very important for the country perspectives, beside regulatory questions, is engagement. I think Claudia really said it all. Engagement is key, and transparency is the key for engagement. We in Mali, within Target Malaria, decided to talk about what we are doing at all the level. National level, we're talking to institutions, we're talking to authorities, we're talking to regulatory bodies. At the regional level, so this is a provincial level, where we talk also to the prefect, to the regional provincial authorities. And we also have what we call the local engagement level. So this is where the lab is on the campus of the university, and where the villages are where we are working, where we are doing the baseline entomology. So almost like every week or every 2 weeks, we have a group to explain the project to. And this includes gene drive. And again, as I said, Mali is not a silo. So this engagement is also global. So, and we participate in the global outreach for gene drive. There is a body independent from target malaria, which is called Gene Drive Outreach. And this is a global initiative, talking about gene drive at the global level. But it's also important that for us in Africa to have maybe a replicate of this gene drive outreach, so that the message is also coming from scientists and regulatory bodies from Africa. The message, I think it's well better received when it's coming from African initiatives. I'm not saying it's not good when it doesn't come from African initiatives, so don't get me wrong. But such an initiative is getting initiated. So we are talking to everybody who is interested to have an African-based gene drive network, so that Africans can talk about gene drive to Africans. And for that initiative, it's not only the engagement. I'm going to dwell into the third pillar also of the perspectives. It's also training, but this time science. Because when we do the engagement, we also get the question, can you do that? Can you guys do the modification in your lab? And not many scientists in Africa can answer yes to that question. I'm probably one of the luckiest one, where in my lab, I have the equipment to do the injection, I have the equipment to do the screening, but it's not true for many African countries. And gene drive, not yet. But genetically modified mosquitoes, without the gene drive, yes. But how many scientists in Africa can say yes? I think that there is an opportunity for training, for bringing the technology into Africa. Having the transfer of technology, the science made in Africa. And I think that will solve the problem of, parachuting technologies. Because he said, okay, phase 0, he called it phase 0. So now we want to bring phase 0 to Africa. And I think that will be very helpful. He also mentioned the co-development. I think that's part of the co-development. We don't intend to say that co-development means that everything will be done in Africa. I think that's not realistic either. But at a certain point, we need to see what is possible, what is not possible. So bringing the technology to Africa will also, help solve another problem with technologies. What is it? Now, we don't have certification bodies for insectaries, and I think that's important. And FNH, I know, is making effort to have such an initiative, so that the insectaries can be certified. And in Africa, we also are thinking about it. We have a Pan-African organization called PAMCA, Pan-African Mosquito Association, where we have started discussion to have such a body to certify insectaries for the technologies. So I think there is an opportunity here. So I have only 25 seconds left. I will finish by the most important part of the question, funding. So I think funding also is important. We need support. Any single African country will not be able to do this because I'm going to say this. I don't know what's going to happen when I get back. But there is no funding for science. And then we need support for that. With that, I will thank you very much for your attention.
Brilliant. Thank you very much, Mamadou. I think we're going to take a few questions now. Again, please stay calm. When Africa Union established the high-level panel on emerging technologies, they selected 10 technologies to look at, and they said we will deal with 3 in the beginning, and one of them was gene drives for malaria control, another was microgrids, and I'm forgetting the third one. You know what that was? The third one was drones. And they asked us at the time, can you do it? And we said, no, we can't. They said, okay, go build the capacity. So actually, the statement that came from the Africa Union, which I believe is in the last slide of Stephanie's presentation, clearly says, as a basic, functionality, please build capacity to be able to talk about this as experts. Don't shy away from it. And I think this is a very strong message from the Africa Union. This panel was chaired by Professor Callistus Juma, God rest his soul in peace. He unfortunately passed on already a few months ago. But they have left us with the responsibility. They have left us with the responsibility to build capacity, to be able to tell our regulators what is the true status of gene drive. Can we use it safely? Is it good for us? And should we go that direction? And we believe that in the wings of people like Abdoulaye Diabate, Mamadou Coulibaly, Kayondo, Prosper Chaki is leading PAMCA right in there. In the wings of people like this, we will be able to answer yes to many of these questions and really support the continent and all our regulators. So ladies and gentlemen, in the spirit of partnerships, we also need a lot of your ideas. We have 2 microphones here. We would like to hear your views. And then my colleagues in the panel will help respond to these ones. Peter Sangoro, I'm going to request you to help us move the microphone around. But please, there is a microphone over there. You can just stand behind and do that. Thank you very much. I'm sorry I gave you a job.
Thank you, Chair. Just one question and one comment, maybe. I'm not too sure which side to start. But let me just ask a question first. The gene drive, where would we recommend this? For high burden countries or for countries that are very close to elimination? And in saying this, as I follow the discussions, I also noticed that we're using the 2 words control and elimination interchangeably. But in some settings, they don't quite mean the same. So trying to put back to the question that I was asking to say, would we recommend this for control settings or elimination settings? Now coming to the comment, I think going to you, Fredros, I think when you retire, you'll make a very good politician. And of course, saying this, I'm also saying politicians make bad decisions or statements sometimes. I think I'm trying to follow the statement that you made about the IRS and the use of liens or mosquito nets to eliminate malaria. And I was a bit uncomfortable when we said we can do all these, but without gene drive, we cannot eliminate malaria. And I'm saying, I'm not too sure about that. We've got examples globally of places that have eliminated malaria without gene drive. And I come from a country that has recorded only 522 cases of malaria in 2018. And there's no gene drive. The main intervention is IRS. And I'm saying to the colleagues, especially from Africa, that if you do IRS and do it well, we can eliminate malaria. Thank you.
So, thank you very much. What's your name again, sir?
Davis.
Davis. From Botswana. Yes. So thank you very much, Davis. I think what we're going to do is take multiple questions and then answer. Very quickly, in response to David, we do agree completely with you. We believe, and our experts have also told us that yes, indeed, there are multiple cases where we can use existing interventions to do a very good job. There are also a number of cases in this same continent where either because of health systems or because of infrastructure differences or just because of the level of transmission that we have at the moment, that we will indeed need new tools. But I think this is a discussion that we must continue to have, and it's really nice that people like Davis are bringing this up. So I will park that question for my colleagues to respond, and we take additional questions. Yes, please.
Hello? Yeah, okay. First of all, thank you so much for this panel. It's been very, very interesting. So I'm Flaminia Cateruccia from the Harvard School of Public Health in Boston, and I'm interested in the safety aspect and the safety standards that were mentioned a few times by different panelists. And so I wanted to ask whether there are actually a set of minimal like safety controls that have been recommended, and what are those? I'll just give you an example, maybe, so that we see, for instance, we work on mosquito reproduction in my lab, and we do modifications that reduce the reproductive output of female mosquitoes. And we often see that when we do that, those female mosquitoes are actually better at transmitting parasites. So, but in a weird way, so parasites don't proliferate more, but they actually go faster. So they are transmitted in a shorter period of time, which would have detrimental effects on transmission. But still, so there have been a few suppression gene drives that have been published, but I've seen no follow-up studies measuring the ability of those females to transmit malaria. And I just wanted to know whether, in order to build trust in the technology and bridge the technology and people, whether something like that is in the plan.
Let's take one more, and then we answer all the three together. Next question. I think we can bring this to the... Oh, yeah, we have one right there.
Hi, thanks for the session. Looking at the end of the development pathway that you kind of laid out for gene drive technology, there seems like an assumption that with all the good community engagement work and all the whole process working out, that everybody's going to be on board for large-scale open releases. But what happens if a region, a province, a country does not agree to this? Then how much buffer do you give them? Can their neighbors no longer participate? What kind of distances do you have to disable for gene drives per se in order to respect their autonomy, their wishes? Thanks.
Thank you very much. I'm going to request one of the panelists or two to respond. Claudia, I believe there's a question that's addressed to you. On the public engagement side, the last one. I think the second question I would like to ask Steve and Mamadou to try and have a crack at those. On the safety concerns, especially how we ensure transparency to get all the safety concerns resolved before moving further.
Okay.
Go ahead.
On now? Or just shout? Okay, great. The first question was about, do we do this technology for control or elimination? And I think for me, for the first testing of first in class, you go into an area where you've got continuous vector populations rather than patchy ones, which you might expect in places of elimination, because you want to drive the construct through. So I think what you're looking for is reasonably
Should I just shout? It's gone off again.
You're hearing me now? Aha! I can hear this falling this voice booming out across the audience. The question was control or elimination. Where would you use gene drive? So I think my answer to that is quite clear. When you're starting out on this, you want to drive the genes through populations. So you want continuous, spatially, populations of vectors to ensure that you get good drive. I think if you're doing it in positions of eliminations, you're more likely to have patchy distributions of vectors, and then you won't get effective spread. I would imagine, at least to begin with, before you know how well it spreads. I've lost myself again. Okay. This one? Yeah. Safety standards. I think that's another lecture or discussion. But I think if I can just raise some of my own concerns, as with any intervention, one measures adverse events. Not expecting to find them, but that's what you do. So you might be particularly concerned about allergic responses as well. You may be concerned about certainly impacts on pregnant women, so you monitor all these things. We've just done this recently in a trial of a new type of net in Burkina Faso, so that's standard. If you're introducing a mosquito which is refractory to malaria parasites, it's me. I'll just bellow, I think.
You can come and use this, Steve. Yeah, come for that.
If you're spreading mosquitoes which are refractory to malaria parasites, there is a possibility you may be selecting for parasites that aren't taken out by those antibodies, the oocysts and the sporozoites. So you may select for other strains. That needs to be considered. There's the issue of impact on biodiversity. What's the role of mosquitoes in pollinating if you collapse the population? Is the construct going to become resistant over time? Is the drive going to come adrift from the effector mechanism? So there's a great deal of things to think about. All part of the risk assessment, and I'm sure there'll be many folk in the audience that have concerns of their own. So this sort of process for risk assessment, I think, needs a hell of a lot of greater thought put into this, so that we measure the right things in the environment, the sensitive things in the environment and in the human population as well. Mamadou, would you like to fill in all the gaps I've left?
Yeah, thank you for the questions. First of all, I just would like to make sure that Ms. Davis is very comfortable. I don't think the intention was to really say that gene drive will do it all. Definitely not. We have experiences that, in the 50s, that IRS, has played a very central role in malaria eradication in some places. So that's really probably the way we crafted our sentences. But gene drive is going to be a very important tool for, in addition to what existed. So that's our thinking, our belief. So now for the question relative to the safety, we have plans, and one of the questions is, we probably don't want any modified mosquitoes being able to transmit more or better malaria than the non-modified mosquitoes. And within Target Malaria, we have a plan to look at those issues, and we are thinking about even some of the diseases that not primarily being transmitted by Anopheles, So those questions, yes, we are taking care of that. And you can think of other safety questions, like Steve said. So I think the answer to the question is yes, we have a plan for that. But I'm not saying that everything is perfect. So there is unknown, there's known unknown, and there's unknown unknowns. So and we are aware of those ones also. So I think there is a question about the transboundary issues. I don't know, maybe Willie will probably take on that. But there are sub-regional regulations, sub-regional policies. The Cartagena Protocol is one of them. So countries that are neighboring, agree on those policies. But if there is a disagreement, I think there is a need to talk more. And the question is very important, even not at the country level, actually even at a village level. You may decide to release in a village, and the neighboring village will say, we don't agree. But again, during our engagement work, we also ought to understand the decision making of those countries or the decision making of those villages, and then we go by that. So that's what I can say. Maybe Willie wants to take on that question for the gap, filling the gap, the transboundary questions.
Yeah, just before Willie, I'm wondering if we have additional questions so that we can add to this. Willie and Claudia will also respond. So, yeah, yes, please. Peter will give you the microphone.
So I am wondering if you're considering modeling conflicting interventions like insecticide spraying, because you will be affecting the gene drives that you'll be integrating into to control the mosquitoes, either replacement or suppression.
Yes, any other question? This one over the other side. While we check that, the answer is yes. All the modeling exercises that are taking place right now consider the fact that there's already a baseline intervention that is either a bed net, an insecticidal bed net, for example, or indoor residual spraying, and that gene drives are being implemented as additional complementary interventions. My colleagues.
Are you considering the effect that you're going to have on the mosquitoes, the gene drive mosquitoes?
Right.
And you might have to drop down one of the interventions.
I get the point. I get the point. There's an elaborate response to this in the sense that you must introduce your construct into the local population. In the beginning, I remember when we started the discussions in this, there were concerns that, what about if the mosquitoes in the village are already insecticide resistant, and the mosquitoes with the drive are susceptible? Aren't they all going to be killed by the bed nets? You get the point? Yeah. So these are genuine concerns, and this is why, for example, we have to think about what are the options of introducing the drive into the local mosquito population so that we are not introducing anything new. But all this information must be put together to generate a joint, position about that. So, yes.
Yeah, thank you. I'm wondering if, with respect to the idea of novel control strategies or strategies for elimination, if there's any room for reconsidering larviciding as part of integrated vector management strategies, which of course there are new technologies that are quite good, including geospatial mapping for predicting sites for breeding, and molecular technologies for reducing larvae in their natural habitats and source reduction. Those have been historically very good at reducing and in fact eliminating malaria in many places. And I don't hear that being talked about as much. So I'm wondering what the role potentially for those are, which may, and I'm only saying may, face fewer regulatory hurdles because those technologies are being used in many places for control or elimination of vector species. Thank you.
Thank you. Last question. Oh yeah, right in the middle. Good. Sangoro, right? Yeah, exactly. Steve, you're gonna take.
Hello. I had a question about, I know you were focusing on Gambi, but I was thinking back on something that Dr. Coulibaly said about biting behavior and whether there's any concern about not targeting like Arabiensis or Finensis that might overtake that niche that is vacated essentially by Gambi once you get rid of it.
Brilliant. So I think we take all these three questions. First, I'm going to put the ball into Willie and Claudia's hands. And then, Steve, if you don't mind, you can take the question on larviciding. And I will ask Dr. Coulibaly and Stephanie to take the last question on Anopheles funesters. Please use this.
Yeah, thank you very much. First of all, I need to emphasize here that approvals are science-based and dependent on case-by-case basis. So if you are thinking suppression or replacement, those are factors that have to be considered during the decision-making process. And the Cartagena protocol is very clear that it has to be a science-based process, though socioeconomic considerations are very important, that my colleague here, Davis, could be alluding to in terms of political and social requirements are very important also during the decision-making process. And therefore, data is very important. So you have to supply adequate data to the regulators. This is safety. And this should also be based on ethical and regulatory standards in those countries. For standard makings, for Camila here, it's mentioned that standards has to be developed. Currently, there are limited standards. However, countries are coming up with this now. But also we have the element of institutional biosafety committees in your organization that should be responsible for also developing some of these guidelines and standards to support your projects. Mamadou has rightfully said for countries, some countries may say no to the technology, but because of the existence of the Cartagena protocol, countries have signed, to ensure that there is a harmonized way of discussions during transboundary movements. There are also regional mechanisms that are also being developed. I talked about NEPAD's role in harmonization process. Regulatory agencies are also in discussions through regional blocks and all that. But if you have a very transparent process as outlined by all of us here, I think the issue of transboundary movements may be minimized. So I think that's what I was asked to talk about. So modeling data is also very essential when you're providing your overall dossier to the regulators. So big data, a lot of information, a lot of public awareness and participation is very important in the whole process. Thank you.
Can I pop up for 1 sec? Just go to address Flaminia's question again. One of the, I think, important recommendations that came out of this was the need for an independent external risk assessment before each phase of expanded testing. And that very important no-go decision point between moving from the lab to the field. So one of the things that we benefited from was having some people who had experience with containment and release decisions for exotic biocontrol agents. And the recommendations contain a fairly extensive list of things that you need to look at to make that go-no-go decision, including things like vector competence and changes in fitness and behavior and that sort of thing. So we think that's very important. Obviously, we tried to be as comprehensive in considering it as possible. But the other thing was that we felt that there needed to be an external risk assessment, not just the team, who may bring along some team bias as to whether they think it's safe or not, but to have an external group looking at those questions.
Okay, So if I understood the question from the gentleman correctly, was about the nature of engagement when it comes to this question of transboundary movement. And Willie already spoke quite a bit about that and the need for regional discussion, which is what's happening. NEPAD is leading this in terms of regional collaboration and harmonization. But ultimately, this comes down to a question, so engagement and ethics, right? So, you know, I mentioned that engagement is reciprocal. So, you know, as a sovereign country, I have a responsibility to engage with Mamadou and with Steve and say, this is what I'm planning to do. And the ideal situation is you would have a regional cluster harmonization, recognizing that the benefits and the potential risks are going to be collectively assumed in this particular region. But at the end of the day, let's say that Steve is completely and vehemently opposed to this and is, you know, is concerned about this transboundary movement. Then the question becomes one of, you know, as a sovereign country, it's the obligations that I have. So I'd have to assess the risks and benefits of, you know, potentially introducing conflict or ill will with my neighbors, and the obligation that I have to say the citizens of my country, if I have a tool which I know is now determined to be safe and effective, to address a pressing public health need. So I think the key thing is that engagement, especially at this regional level, is reciprocal, and we have responsibilities. I have a responsibility to engage my neighbors, but my neighbors also have a responsibility to engage with me and understand what those risks and benefits are, because potentially you're standing to gain and to assume the risks collectively.
Thank you. We are 2 minutes beyond time, but I think there was a question about larviciding and why we are not, whether it's still a good idea to consider these are the tools that are known to be effective. I'm going to ask Steve to try and tackle that. In addition to what Claudia said, at least in Africa, we are lucky to have the NEPAD agency, which is already spearheading together with the regional blocks a series of training opportunities for regulators, but also trying to create an environment where regional blocks can uniformly review the technology and make a decision as to whether it is a go or a no-go, really just being transparent about it. So, Steve, if you can tackle the question on larviciding, and then we should be coming to a close.
Thank you. I'm actually going to make it more broader than that. I think one of the important things is that we don't oversell this technology and that people wait for this technology. We are at a critical phase in the history of malaria control in sub-Saharan Africa where control has stalled and is increasing in some countries. And I think it's really paramount that we continue to deploy nets and IRS on a large scale. And yes, sir, I agree absolutely. I'm a great fan of larval control. This is an important additional tool. And we're looking for other interventions outside the health sector. So as much as we can do. And then to address really the issue about, well, how can you work out whether gene drive works if we're spraying everywhere? Well, I think you don't go to areas where they're doing heavy spraying. I think site selection is critically important. But don't take your foot off the gas, I think is my finishing line. Thanks.
So, ladies and gentlemen, thank you very much. I'm going to ask my co-chair to close the meeting.
Thank you, Fred Russ, for that excellent moderation, and thanks to all the panel members for their great contributions, and thanks to all of you for coming. I hope that you learned something and that you are walking out of this meeting feeling a little bit more confident about the way that this new technology is being pursued and how carefully it's being considered. And we thank you and enjoy the rest of the meeting.
Hey, good morning. I'm sitting at these friends. How are you doing?
Hey, how are you? It's good to see you. So.
Potato getting colder, right?
I hope so. I don't know. This is, you always expect the unexpected with this one. Yeah. Yeah, good.